And good morning, everyone. Welcome. We're delighted to have you join us for today's webinar from Regenxbio, who will be sharing their positive top-line results from the pivotal phase 3 AFFINITY Duchenne study with RGX-202. I'm Michael Kelly. I'm the Chief Scientific Officer at Cure Duchenne, and I'm going to get us started with a bit of housekeeping. First, you'll have an opportunity to ask questions today. You can simply click on the Q&A button at the bottom of the screen and submit a question anytime throughout the presentation. We'll have a moderated Q&A session at the end of the webinar, and I'll be joined by my colleague, Dr. Liana Orlando, Cure Duchenne's Vice President of Research. For those questions that we may not be able to address or get to, we'll work with the Regenxbio team and follow up. And finally, after today, this webinar will be available on Cure Duchenne's website as a resource for the community. So let me begin. Regenxbio's approach to gene therapy as a treatment for Duchenne utilizes a proactive, short-course, immune-modulating regimen designed to improve safety, while delivering a unique microdystrophin gene construct that has been specifically designed to include an extra feature of the important C-terminal region of the gene. Consequently, the RGX-202 microdystrophin is larger than other microgene constructs, and their clinical trial is being closely followed to understand how the enlarged microdystrophin gene and the immune modulation protocol perform in patients. The company has just released their long-awaited top-line results from their fully enrolled pivotal phase 3 AFFINITY Duchenne study. And to learn more about this, we have Dr. Naz Dazgir, the clinical development lead at Regenxbio, with us today to talk about the findings. So without further ado, let me hand it over to Naz to get us started.
Great. Thank you so much, Mike. I appreciate the introduction, and thanks to everybody for joining on this webinar. I hope you enjoy this nice May day, and we'll get started. So we're going to be talking about RGX-202 as an investigational gene therapy for Duchenne muscular dystrophy. This is an update of our pivotal data from the AFFINITY Duchenne study. These are our forward-looking statements. All right, so I'll tell you a little bit about RGX-202. Some of you in the crowd may be familiar with this already, so this is just a rehashing there. RGX-202 is an investigational one-time AAV gene therapy designed to address the lack of functional dystrophin. So we deliver our drug with an AAV8 vector. As you know, there are many different types of AAVs that can be utilized as a vector, and we use AAV8. We also have a unique promoter that is called SBC512. That is a promoter that's really helps our transgene get to the correct location and kind of express the protein in the correct location. And we have our microdystrophin gene, which is the transgene. This is a synthetic gene that was developed to deliver what we thought were the best components of the dystrophin protein. So basically, the dystrophin itself is too big to package into the vector, so you have to kind of pick and choose what are your critical components that you'd like to include. And the one critical component that's highlighted in reddish orange here is the C-terminal domain that we included in ours. So as I mentioned, the vector is designed to deliver the transgene to skeletal and heart muscles in the body. The SPC512 drives the expression of the transgene in the muscles, and the microdystrophin transgene retains the key elements, so not the whole dystrophin, but key elements, and that includes the C-terminal domain, which is highlighted here in this figure on the right. And that C-terminal domain, which is also in red in this figure, kind of binds to this kind of gray area that spans the muscle membrane, the cell membrane there, called the dystrophin-associated protein complex. And we hope that that actually anchors the transgene and further stabilizes it to the correct location in the muscle cell. The CT domain has also been shown in animal studies to protect muscle and improve the muscle's ability to repair itself. The drug here highlighted with this IV bag is given via a single IV infusion, and immune suppression and antibiotic medications are also given prior to and for several weeks after receiving RGX-202. So this is just a demonstration of what I talked about earlier. We have the full-length dystrophin protein on the left and the microdystrophin on the right. And you can see there's a few less circles here, but that's what we can package into the vector itself. But we do have some of the critical components, and highlighting here again, the most unique component of our transgene is this C-terminal domain. And these are the preclinical studies, which means these are mouse studies. This is before getting into humans. And they demonstrate that the CT domain in RGX-202 microdystrophin preserves skeletal and cardiac muscle health and enable muscle resilience. So how does it happen? So we see prolonged microdystrophin activity. So the CT domain significantly enhanced restoration of the dystrophin-associated protein complex by stabilizing the muscle microdystrophin in muscle cells and potentially allowing it to accumulate at higher levels and act for extended periods of time. It also protected against contraction-induced damage in the DMD mice and enables for better muscle recovery. So if you look at the figure on the right here, this is a figure that shows percent initial force generated by a mouse with a contraction, with a grip, and then basically the amount of contractions that are performed. So the initial grip might be really strong, but then if you repeat it after 20 times, it weakens over time in the Duchenne mouse in particular, which is shown in this darker gray. In the control mice, you can see that that goes down, but not by too much. It's like at 100%, and then it kind of fatigues around close to like 60%. This blue here shows what the DMD mice with the microdystrophin that we had without the C-terminal domain did. So they also decreased, were a little bit better than just DMD mice alone without microdystrophin, but when we added the C-terminal domain, we noticed a significant difference in function, and that supported our decision to move on to include the C-terminal domain in the transgene that was designed by our company. Additionally, in cardiac muscle health, RGX-202 microdystrophin protected against cardiomyopathy in a pharmacologically induced model. So this was, again, inducing some toxicity to the heart muscle in the mice, and then testing recovery of heart function in the mice. And we noticed that the C-terminal domain in the same similar studies performed in the mice also helped with recovery there. This is highlighting the immune suppression regimen that I talked about earlier. So the top figure here starts, the D's are days and the W's are weeks. And this kind of highlights, it's actually extracted from a paper, this image, that highlights what's been described since gene therapy has been started, what we've learned a lot about viruses in general from gene therapy, and the immune response. So the amount of a viral load that you get with gene therapy is actually really unnatural. You wouldn't get this in just regular exposures to patients, or you could actually get AAV in the wild, but you would never get it to this amount. And what we've learned from other therapies and what we've taken from this learning is our immune suppression regimen. So basically, day 0 is your exposure to the drug. And within minutes of exposure, your immune system starts to respond to the drug that's administered. The first round of events that occur with your immune system are what they call complement-mediated events, or they're part of your innate immune system. This is when your complement goes crazy, it goes ballistic, it starts firing, it's not targeted, like soldiers on the attack, but not knowing where they're shooting, and they go crazy. And that kind of peaks around day 6, day 8, and then it tapers out over time. Just as the innate immune response is tapering down, then you have your adaptive immune response kind of leading the charge. And the adaptive immune response is more strategic. They've had time to think, and you start to form antibodies that are targeted to the antigen or the virus being delivered. Now, you can form antibodies to the virus vector itself, or you can form antibodies to the transgene as well, if it's presented in the muscle cells, for example. So the way we designed our immune suppression regimen was, one, to address the complement activation. And that is with the use of eculizumab. So you'll see the timing of the eculizumab is we have 5 doses total. We start on day -9 and we complete on day 12. This is to address the innate immune response with the complement activation that can trigger sometimes severe thrombocytopenia or thrombotic microangiopathy, and liver injury, sometimes even cardiac toxicity. Thrombotic microangiopathy is kind of like a inflammation of your really tiny blood vessels in your body that then trigger platelets to get to that spot and start kind of clotting up little microvessels in your body that can then lead to kidney failure. And that could be fatal at times. So that's why we took it very seriously based on what we'd seen with prior gene therapies. And we instituted eculizumab. And eculizumab is a complement 5 inhibitor, a C5 inhibitor. It's a little bit downstream from other complement inhibitors, so it doesn't completely shut down your complement system, but we find, knock on wood, with our good safety profile that it inhibits it just enough to give us a good safety profile. The sirolimus is also instituted to inhibit B and T cell activation, and this is to address the adaptive immune response. mTOR is mammalian target of rapamycin. And basically, the sirolimus kind of works to prevent a lot of antibody production and also prevent the antibodies from kind of turning from one type of antibody to another. And we find that that would be very helpful to allow for less of an adaptive immune response. There are speculations that sometimes inhibiting this adaptive immune response can also allow for persistence of the vector, but these are all speculations based on animal models, nothing that we've proven in our study at all. But does it theoretically also give a chance to our drug to get into more cells because it's not being attacked and shut down that quickly? Speculation, but something that's a theoretical benefit. And then we also have the additional steroids. That's pretty much given across all gene therapies. That starts on day -1 and continues for a full 8 weeks after dosing and then tapers off weeks 9, 10, and then 11 and 12. Sorry, I forgot to mention that sirolimus starts on day -7 just to get that level therapeutic, and then you come off of that afterwards. You stay on the sirolimus for about 8 weeks after dosing and then wean off on weeks 9, 10, 11, and 12. With the eculizumab and the sirolimus, you also have to go on to antibiotics because, of course, immune suppression leaves you at risk for certain opportunistic infections. So you do go on a specific antibiotic for eculizumab, and you do go on a specific antibiotic for sirolimus. And additionally, for the eculizumab, you will also be asked to get a meningitis vaccine if you have not already received it. So I went through a lot of the details here. This is a little schematic just to kind of hammer it home. So you get your viral gene therapy, your immune response is triggered. We have that innate immune response. That's really the complement system being activated. And then you have your adaptive immune response. And basically, that's the antibodies that are driven by the B cells and the T cells. And the eculizumab targets the complement system. The sirolimus targets the adaptive immune response and shuts down the mTOR go signal, and basically meant to address the issues that we talked about before. So the complement system puts you at risk for thrombotic microangiopathy, thrombocytopenia, or the low platelets, liver injury, or acute cardiac injury. For the adaptive immune response, which we didn't highlight on the prior slide, basically the antibodies either to the vector or to the transgene can cause myocarditis or liver myocarditis. Typically, it's antibodies to the vector that can cause liver injury. And then typically it's antibodies to the transgene itself that can cause a myositis. And itis just means inflammation. So myositis is muscle inflammation and myocarditis is heart inflammation. And the prednisone addresses all of this as well. All right. So I'm going to talk to you about our clinical study. So this is the AFFINITY Duchenne study. We had a phase 1/2 study that was a safety and dose finding study, sometimes called a first in human study. We identified which dose levels are potentially safe. We had two dose levels, dose level 1 and dose level 2. Dose level 2 is twice as much vector as dose level 1. We gathered some early efficacy evidence to identify which dose level to carry forward to the next stage, and we decided on dose level 2. And dose level 2 was tested in the pivotal study, which is a phase 3 study where we checked safety, biomarkers, and efficacy. And we dosed enough patients in the pivotal study at the selected dose level to understand the safety profile. We also collected biomarker data, which is not functional data from motor function testing, but blood work, biopsy results, and to support potential accelerated approval, which you would get from the FDA. So trying to use the biomarker data because sometimes it takes functional data a while to declare itself, but convincing the FDA that the biomarker is reasonably likely to predict future functional benefit. So that's the whole point of accelerated approval. And we also wanted to collect functional data to evaluate efficacy. And then we've also been enrolling in the confirmatory study, and we're almost near the end. And this is for additional safety and efficacy data. And if we're granted accelerated approval, we'll be able to continue to gather additional safety and efficacy data and dose additional patients. We use this to dose additional patients to confirm if there's any functional benefit. So we still collect the biomarker data in these patients, but that's not the primary goal of the confirmatory study. The confirmatory study is to really hammer home whether or not the drug produces functional benefit as measured by motor function outcome measures, for example. So this is a highlight of the study and the key eligibility criteria. So we enrolled ambulatory boys who were greater than or equal to 1 year of age. They needed to have genetically confirmed Duchenne muscular dystrophy with deletions or point mutation, except those patients with deletions or point mutations in exons 8, 9, and or 10. But we did take everybody else. Unfortunately, due to that region being designated as high risk for now of potential myositis, those patients are excluded from the study. And the patients had no pre-existing antibodies. So in this pivotal portion of the study, we did not have a titer cutoff established. So basically, you're either positive or negative. So anybody who was positive was excluded from the study. The dose, as I mentioned before, is the pivotal dose. This is 2 × 10⁻⁴ genome copies per kilogram. And we had some enrollment criteria that were a bit different for those who were 1 to less than 4 years of age versus those who were greater than or equal to 4 years of age. So for those who were 1 to less than 4 years of age, they needed to be able to complete 10 meters without a 10-meter walk without assistance. They could have been on a stable dose on or off corticosteroids prior to enrolling. They needed to be at least 10 kilograms. It's not due to any safety issues. It's just that we do need to take blood for the study to measure safety, and there's only so much blood you can take. So there's a limit, and it's usually a weight-based limit. So that's why we put that weight cutoff there. And they needed to be able to perform the supine to stand without assistance. For those who are over 4 years of age, they needed to be able to complete a 100-meter walk without assistance, be on a stable dose of corticosteroids for 12 weeks, have a baseline North Star Ambulatory Assessment score of greater than or equal to 16, and have a time to stand of greater than or equal to 3 and less than 7 seconds. And of course, they were all on the same proactive immune suppression regimen that I discussed in the prior slides. Our endpoints, our pivotal trial endpoints are listed here. So the primary endpoint for this study was the proportion of patients with greater than 10% RGX-202 microdystrophin expression at week 12. So what does that mean? So that means that you take a sample of muscle tissue from the biopsy and you can quantify the amount of microdystrophin being expressed on there via a method known as Western blot. This follow-up biopsy was done at week 12 compared to the baseline and compared to the baseline where there was obviously no microdystrophin available. The secondary endpoints included for the over 4-year-old patients, the velocity of time function tests, the North Star Ambulatory Assessment, and safety assessments. For the 1 to less than 4-year-old patients, we did the Peabody Developmental Motor Scale 3, the stride velocity 95th centile for the 1 to 4-year-olds, and safety. And the exploratory endpoints for the over 4-year-olds were the stride velocity 95th centile and MRIs. We did not do MRIs in the younger children who could not tolerate it without sedation because we didn't want to introduce that risk. So this is the interim safety data that I wanted to talk to you about today. So we overall, I'm being completely honest here, I'm really, really pleased with our study. I don't want to jinx it. I feel happy when I go to bed at night that we've been dosing a ton of patients and I don't have to super stress about, I mean, obviously I'm always worried about every patient and I anticipate risk all the time, but I do find that our drug is well tolerated and our immune suppression regimen is a beautiful safety net. Despite that being in place, we did have two treatment-related severe adverse events. They are called severe because the children were hospitalized, but thankfully the children were fine and recovered quickly, which again, I think speaks to the assistance that we got from the safety of the drug and the immune suppression regimen. Both of these events took place in kids who were over 4 years of age, and I'm going to kind of tell you some details about them. So, the subacute myocarditis case was an 8-year-old boy, 23 kilograms at dosing. So, that's about 60 pounds. He had a premature stop codon in exot 60. He presented with subacute myocarditis 33 days after dosing. And basically, he had normal troponin I. So there's troponin I that you can check, and then there's a more volatile high sensitivity troponin. The PI who was taking care of this patient had prior experience with other myocarditis cases in prior experience with gene therapy. And he said, I like to check troponin outside this, like outside the study, like calling for a regular troponin. I'm going to check a high sensitivity troponin, which is completely fine. Do what you got to do. He did that and he saw it fluctuating. And basically, the upper limit of normal is 47, and that child's went up to like the kind of close to 70 or so, which was not high at all and wasn't really concerning. But what happened was that this PI had also dosed many patients in our study. And this kiddo had some, and you'll see, as you can see here with the treatment related adverse events listed, one of the most common things that we see in our kids are vomiting, fatigue, and nausea. So this child had the vomiting and nausea, but it persisted beyond what we usually see, which is about 14 to 16 days. It was like obviously day 33, and he had this episode of vomiting. Very mild pain. And he said, you know what? Just to be safe, I'm going to get an echo and an MRI and work this up. Well, he had the MRI done, and there was not the typical evidence of what you see, which can be late gadolinium enhancement, but some other criteria on imaging that met the diagnosis of myocarditis. So after that, the PI, who again had previous experience, we defer to the PI on the management of the child. He treated the child, and basically the child was asymptomatic, didn't have any arrhythmias, and actually was able to have a follow-up cardiac MRI a year after the event. And that heart function is perfectly stable at 65% on the ejection fraction, and he has no residual scar tissue from the event. So we are so happy that he's doing well. He's functionally doing well, and that was that case. For the liver injury case, that was a 10-year-old boy, 34 kilograms at dosing. He had an exon 3 to 7 duplication. He had no symptoms whatsoever. It was based on labs. Um, the AST and ALT liver enzymes in kids with Duchenne are a little bit hard to track because they fluctuate a lot with the CK, although I think some folks have been doing really good work on seeing if you can do a ratio of ALT to CK to really pick it up. But what we like to look at is GGT, and the GGT was starting to kind of track up in this patient. And basically, for this patient, the PI decided to give some additional PO prednisone, and it was still kind of going up, so she decided to do IV methylprednisolone. And when she made that decision to do IV methylprednisolone, it happened over a weekend. And basically, she said, oh, my infusion center is not open over the weekend, so we'll have to bring him in. And he got admitted, and that's why it became a serious event. But thankfully, he was great. He was asymptomatic the whole time. He got 5 doses of IV methylprednisolone. He had an abdominal ultrasound, totally normal. Bilirubin levels were normal. All of his labs, there was nothing pointing to any cause for this, other than, possibly, related to the gene therapy itself. So, but basically, we were very happy that he resolved after an easy course of methylprednisolone, went home, and he's remained stable ever since. I kind of touched on the treatment events that we see in the kids otherwise, vomiting, fatigue, nausea, abdominal pain. That abdominal pain can come with the nausea. Sometimes the steroids make that with a little trigger that a little bit with the reflux that can happen. And pyrexia is fever. We thankfully did not have any drug-related thrombocytopenia, low platelets, myositis, or neurotoxicity. So in our study, our primary endpoint was met with high statistical significance. The primary endpoint is to support accelerated approval, which is the proportion of participants with a microdystrophin expression of greater than 10%. At week 12, 28 out of our 30 participants, which is 93% of our patients, achieved greater than 10% microdystrophin expression. 80% of the participants achieved greater than 40% microdystrophin expression. If you average all our patients together, the average microdystrophin expression was 71.1%. And we did a subgroup analysis on the older kids, and we were very happy to see that on average in our kids who were 8 and older, we got a 41.6 average microdystrophin expression, which is the highest expression reported in older age groups across gene therapy programs. So overall, there was robust expression achieved across ages, supporting potential for improved functional outcomes. This is just a summary of the biomarkers, as I mentioned, not the functional data, but the biomarkers that we tested in our study. This is microdystrophin levels by Western blot that are listed here, kind of divided into this different age groups, but I'll just kind of highlight the overall numbers here because there's a lot of data here. The next column is the vector copy number, which is copies per nucleus of drug within the muscle cells themselves. And the positive fibers by immunofluorescence are showing how many fibers are actually expressing the protein on the rims of the muscle fibers themselves. Western blot basically quantifies the protein amount, but it doesn't tell you that it's actually getting to the right place. That's where the immunofluorescence actually helps. So it gets the right, we know it's in the muscle cells, and we know it's actually localizing to the sarcolemma. So overall, we had a 71.1% microdystrophin expression. The vector copy numbers were about 19.6, and we had 60.6% immunofluorescence positive fibers. This is our functional data. So our participants exceeded external controls on all functional measures at 1 year. So what I'm showing you here is something called propensity score weighting. This is not a placebo-controlled study. This is an open label study. What we did is we got access to external data sets. We were able to purchase one through Cure Duchenne, so thank you to Cure Duchenne for that. Basically, we put in some baseline characteristics, the baseline characteristics of our patients, and then you can see how those patients, whatever patients matched based on those baseline characteristics, which you can see here, the external control N. This is the number of patients that each of these values is being compared to. And basically, whoever matches, we watch them over time, watch the matches and how they perform over time, and compare them to how our patients did over time. So our N, or our patients here, are listed here. You'll see this one patient is just at 8. That's because he was non-compliant with participating with the assessment, which happens with kids, and it's okay. So VTT stand is velocity of time to stand. So you can see that anything past the dotted line here in the yellow section favors RGX-202. So basically on velocity of time to stand, velocity of 10-meter walk run, velocity of time to climb. And on the NSAA, all of our patients are doing really well and improving compared to what would be expected for their trajectory as predicted by their baseline function. So very reassuring to see that. And this is at 1 year. And then we did an additional subgroup analysis. And you can see we had 9 kids over there, but 5 of those kids were over 8 and older. And even in that group of older patients, at 1 year after dosing, we see that they are performing better than what would be expected based on their trajectory with propensity score weighting on velocity of time to stand, 10-meter walk run, time to climb, and NSAA. And this is a really great slide that shows that microdystrophin correlates with the functional outcome NSAA, or the North Star Ambulatory Assessment. So this is obviously early data. We have 9 patients here, and we were very excited to see this because it hasn't been described here before, but we have statistically significant correlation with the NSAA change from baseline and when we compare it to CTAP predicted value at 1 year. So let me kind of hash this out for you. So the figure on the left shows the change from baseline total NSAA score, so how the kids did over time, and compares it to the microdystrophin level. And you can see that we are seeing a really good correlation here with the statistical method, Spearman correlation, and the p-value between the microdystrophin level and the change in from baseline NSAA. So supporting the argument that microdystrophin can be reasonably likely to predict functional benefit, and this is at 1 year after dosing. On the figure on the right, we also checked to see how the kids were doing compared to what was predicted for them 1 year after dosing. So the difference in the change from baseline of RGX-202 and CTAP NSAA total score. So CTAP is the collaborative trajectory project, which basically is a model. It's almost like an equation of sorts. You input your patient's data at baseline, and then it predicts how the patient's going to do later. It's only available for the NSAA, but you can see that the predicted trajectory versus how our patients did, that difference also correlated with microdystrophin expression. So also very happy to see that. So we're going to continue to monitor this. Hopefully it'll continue to declare itself in this way. So stay tuned. Another thing that's very important to us is how the families perceive how their children are doing. So these are patient-reported outcome measures. This is called the PODCI scale. And in transfer and basic mobility, sports and physical functioning, and global function, the RGX-202 treated patients' families demonstrated that they had an improvement. And what you see in gray is what was predicted after a 12-month period for those same patients. So caregivers reported observed improvements in function in the home and community as measured by the key dimensions of the PODCI at 12 months. So the confirmatory study is ongoing. So we'll talk about where we've been. The phase 1/2 portion evaluated RGX-202 at 2 dose levels. Interim safety and efficacy data enabled moving forward with future studies. The phase 3 or pivotal portion is fully enrolled, and the interim results have been shared with this top-line data set. And RGX-202 microdystrophin expression at 12 weeks indicates that the primary endpoint has been met. There's encouraging interim functional data in the treated patients, showing functional improvement at 1 year. Where we are today, we expect, thanks to our sites and our patients who enrolled in the study, we expect to complete enrollment of the confirmatory portion of the AFFINITY Duchenne study by mid-2026, so very soon. And the confirmatory portion continues to evaluate RGX-202's impact on safety, biomarkers, and functional outcomes. And what's next? So we expect to submit a biologics license application, or BLA, under the US FDA's accelerated approval pathway soon. So thank you so much for your time. And this is how you can get some more information on RGX-202 and the AFFINITY Duchenne trial. And if you have any questions, you can reach out to Regenxbio patient advocacy team. That's the email, duchenne@regenxbio.com.
Nah, super job. Well done. Nice walkthrough of the data. Congratulations on it. It's a really nice data set.
Thank
Let me
you.
invite Leanna there you go, to join in. I was wanting to actually start to talk through the 2 SAEs, but you actually done a great job on per patient level, been able to explain and understand that. So I think I could really rest assured at night as well, that the package actually looks quite good. The question I would ask is, how long are you going to continue to follow those boys quite closely, the ones that had the SAEs?
Yeah, so the main study itself is for 2 years. So obviously we follow them for that period of time. And then there's a long-term follow-up study that goes on for another 3 years after that. That's the duration of the long-term follow-up at this time. I would assume, hopefully, once we get approval, like if the children experience any potential side effects or anything like that, that would enter into a FAERS database and folks would always be very involved to continue to monitor and study. I take patient safety. I mean, I know everybody in the space does,
Mhm.
but I'm kind of like obsessed with it. So I like to kind of study and make sure that we do what we can to identify risks, future exposures, things like that that could potentially trigger these events and how to avoid them and whatnot. So, we have the patient's best interest in mind and we'll always be available for them if something comes up.
Okay, thank you for that. Can I
Mhm.
ask, when do you anticipate sharing some of the data from the young population, those from aged 1 through less than 4? I'm particularly intrigued to see how this particular gene therapy is going to affect those individuals.
Yeah. I think that's kind of not my decision on when we're going to be sharing that data.
Hmm.
I think there's a concern because every time you look at your data, you take a statistical hit. But I think if we were going to do another data cut at that time, we will have had like a good number of younger patient data. That being said, our youngest patient dosed, I think, was around like a year and a half old. So even if we got a year's worth of data,
Hmm.
I mean, we'll see what the SV95C does. I mean, it's hard to have them differentiate from the pack at that age, is what I'm trying to say. I I think that that's why we're kind of seeing a really nice positive change in our 8 and older patients too, is because they're also like deviating from what you expect.
Hmm.
I know that some of the children with Duchenne have been identified now as you've been more in that space as having early developmental delay. So we're looking with the Peabody Developmental Motor Scale to see that they can normalize to what would be their expected trajectory. But funny enough, my daughter, when she was in kindergarten, had a little boy with Duchenne in her class, and I would be able to go there to the playground and watch him play. And man, I mean, at that age, I think he was 4.
Hmm.
He was climbing up and down the slide. I mean, it's hard, right? He was very active, you know? And I think it's really hard to differentiate these kids from the pack, except when they hit that decline phase, which is when they're 6 years old. But maybe the SV95C can pick up on that for us. Maybe the Peabody normalization can pick up on that for us. We haven't looked at that data yet, but it will be interesting to see, and we might be able to provide that. But I'm not totally sure when we'll
Yeah, I
provide that.
agree. Like for me, I was particularly struck with the Spearman correlation, with function and the amount of transgene that's in there. And it's clearly mapped out quite nicely, in the data that you shared. I expect that's going to have some sort of similar correlation in the younger patients, and
I hope
that's going to
so.
be, yeah, I think it's going to be a really rich data set to go over whenever you guys are ready to release it.
Yeah, I hope so.
Yeah. Liana, I don't want to leave you out. I see you there in the corner.
Yeah, no, thank you. And since you mentioned the Spearman correlation, I'm going to start with one of the most sophisticated questions, actually. We can talk about microdystrophin in general. So,
Um,
um, you know, so well, we'll start small. First of all, is there plans to measure microdystrophin by anything other than Western blot? Is one of the easy questions that came in. Maybe you could speak to.
um, so early in part one, we did have Western blot and LCMS, which is mass spectroscopy, which is another way of measuring it, but we just saw that there was a really good correlation in results and pruned that down to just being Western blot for the pivotal and confirmatory study.
Great.
So for now, that's all we're doing for microdystrophin.
Perfect. So to get back to that correlation of the microdystrophin expression and the outcomes, there's a question here which is quite sophisticated, was saying at the baseline function of the individuals, was their NSAA a predictor of microdystrophin expression? So said, in another way, whether, the individuals who had better baseline function ended up taking up more transgene, having better expression, and whether or not that could be another interpretation of the data.
Great question. But when you look at this other slide here on the right, it kind of shows the prediction for someone with that same baseline based on CTAP versus how the 202 children are doing. So that shows that there is a difference than what would be predicted by their baseline function. And I would argue, I mean, it makes it a little difficult if the kids are a little bit better functioning, because then you have a little bit of a ceiling to how high you can go with the NSAA, and actually you get the more dramatic improvement with the kids with the lower NSAA, but not too low. There's that sweet spot that you want to capture to treat.
No, that's wonderful. And speaking a little bit about the data sets, because there's some questions that came in from that, asking like, how old were they? And I know that you've used multiple data sets, correct?
Oh, yep.
And even for like that in the CTAP model, you end up selecting from the data sets the individuals who sort of match the profile of, So
yes,
some
totally.
of the variability
We don't have that. We don't have that slide in here. I think we kind of took it out because it's a busy slide. It is on the other deck that was, I think it's on the deck that Dr. Panda presented at ASGCT. But it has all the baseline demographics of the kids and whatnot. And also the methods, the statistical methods by which we did this. But basically it's age, time function tests, NSAA, and steroid use. That's how we match them. And then there's a weighting process for the propensity score weighting. CTAP it has its own thing. I think if I remember, it's like age, baseline NSAA, maybe, I don't want to misspeak, but maybe BMI is in there too. I forget, but we send it to them to do the analysis and then get the results back.
Great. Okay, so switching gears a little bit, I'm going to go back to the beginning maybe and talk a little bit about the immune protocol. Because there's a lot of questions that came in about that. So just in general, like how does your immune suppression protocol compare to others? And this person even mentioned actually even for other standard methods for like organ donation.
What's the question for organ donation? Sorry.
Well, how does your immune protocol, compare to, the way I read it was these drugs are used in other uses, right? With the safety profiles associated with them.
Yeah. So, okay. Our immune suppression protocol was, I think, the only one using sirolimus before. I think a couple companies have started to use it, not for all patients, and some people commercially use it as well for gene therapy right now. So that's becoming a bit more common, although not all gene therapies obviously use sirolimus. They just mostly use prednisone. The eculizumab is pretty unique to us, I would say. I think maybe Rocket Pharma uses that for their Danon disease study, but I'm not totally up to date on their most recent treatment regimen. So that's unique to us for sure. In terms of the safety profile, obviously eculizumab, there are some concerns about meningococcemia, and giving the combination of antibiotics and the meningococcal vaccines is every step needed to try your best to avoid that from happening. And even with that, the risk is really, really low. I know people get very nervous about it. I will tell you that eculizumab is a reversible drug, and it gets out of your system pretty quickly. It is not like other immune suppressing drugs like rituximab, for example, that can cause potentially in some cases like persistent B cell suppression. This is a reversible complement inhibitor. So those people who use eculizumab all the time are very comfortable with it. Those people who do not use eculizumab are very scared of it. But we've been using it. We've been grateful for it. Would it make our lives easier to not have to justify using it all the time? Yes. It's a question that comes up all the time, but we are so pleased with what it's doing for us. And we are grateful for this drug because we take this process very seriously. And we don't want to lose a single patient. So that's why we take these measures.
Yeah, Leanna, may I expand a little bit on this? Go ahead. I'll jump in after you.
Okay, well, I was gonna say that the other piece of it, which I think is really different than organ donation, is individuals aren't taking then these drugs chronically for the rest
They're
of
not.
their life, right?
No.
There's a period of time, exactly.
But the organ donation thing, I'm very confused by, but very intrigued by whoever asked that question,
Mhm.
because interestingly, our consultant said that eculizumab is actually used off-label for many things, off-label. Um, and one of the things people have realized is that when people pass away, there's a huge inflammatory response. And they've started to use eculizumab in that scenario so that the organs can be harvested without crashing in that huge inflammatory response that happens with death. So I
Interesting.
don't know if that's where the question arises from the organ donation part, but
Yeah.
that is something where eculizumab comes into organ donation, actually.
Yeah,
Mhm.
and just building upon that, I think there's a number of questions which are sort of questioning, are the adverse events that that are reported related to the immune suppression regimen,
Oh,
or and I think, yeah, go ahead and clarify.
you mean the SAEs that we had? Um,
Or yeah, or even any of the related
oh,
adverse
okay.
events. Yeah.
Yeah. Okay. So the SAEs that we had are not likely related. I mean, I think I would have to go back and see if they were listed as being possibly related, but people put, if you're in clinical trials, you'll see like, oh, it's possible, it's possible, it's possible.
Yeah.
But I think more probable that it was related to 202 was the case with these. With the immune suppression regimen, I would say most kids hate the taste of sirolimus. They do get abdominal pain and nausea with prednisone. Eculizumab is tolerated like a gem. No issues there. Our biggest offender is probably the Bactrim that you have to give with the sirolimus. And that's why because we've had a few kids that have had unknown sulfa allergies and can get an allergic response because they've never been on the drug before. So then we just have to switch their antibiotic.
Mhm.
And they've been fine. They get a rash.
Go ahead, Mike. You wanted to jump in?
Yeah, just expanding on it a little bit, actually. Um, Now that we've seen those two SAEs and understand essentially the extent or the non-extent of both of them, does that give you any confidence moving forward? We're about a year or more away from this drug being approved. Thinking about those patients with exons 8, exons 9, or 10 deletions getting access to 202?
Yeah. Um, I personally have a lot of faith in our
Mhm.
regimen, and that is a risk that is our company's decision to take. But personally, as a clinician, not as someone working for Regenxbio, I would say based on what I've seen, I would be comfortable taking that risk.
Yeah, there's a lot of families out there that would say exactly the same, I can say. And also in a related manner, the exclusion criteria was any antibodies to AAV8. And
Yes.
again, you're about a year or more away from this being commercialized in a different setting. Are you thinking about relaxing that where you want zero antibodies or to 1 to 200 or 1 to 400? Like, what's your thinking or guidance around that as we go through the next year?
Yep. We would definitely like to change that criteria. We've worked to kind of expand that to be less than 1 to 50.
Hmm.
Um, and that got implemented, but we have not yet, to my knowledge, dosed anybody with that.
Okay.
Everybody's been less than 1 to 20. But I think future plans are to, yeah, definitely dose people with or attempt to dose people with higher. We check we check total antibody,
Total,
not
yeah, total
neutralizing
binding
antibodies.
antibodies. Yep.
So we would be checking that and hoping to dose kids with higher, but you would have to do a whole study, a safety study, to be able to proceed with that. So I
Okay.
think not with this first round of approvals, but I think the goal is to, in future studies, test the safety of this to make sure it's good.
Yeah,
And
and
also the efficacy. You
yeah,
don't want the drug to be demolished as you give it.
I totally agree. I think there's a possibility of taking a hit on that, but I think the communities there are asking those types of questions, and they're thinking about next year this is available and who's going to be the population.
Yeah.
Mhm.
Yeah,
Yep.
maybe we could reiterate that you're both absolutely correct. Lots of people are asking, what's happening right now with the trial? Are there still spots? And then, when it's available commercially, what are the estimations about who might qualify for this? What would be the requirements?
Great question. In terms of enrollment, I would say, I think we're pretty much almost there. Like I said, mid 2026, we will be done. I would consider June to be mid 2026. So, obviously we're not fully enrolled yet, but by that point, I think all the kids that are going to be dosed are already being screened, at this time. Um, and in terms of who would gain access to the drug, I think that's going to be a question about what the FDA says
Mhm.
and also what insurance companies are willing to approve.
Yeah. Can I just
Um,
expand a little bit on that question? Not to
sure.
put you on the spot, because I know the
Yeah, yeah.
FDA discussions are ongoing, but I imagine that you're going to continue to file this year with an approval at some point next year. That's still the company strategy?
Yep. We have a rolling BLA at this time, and our goal is to aim for an approval next year sometime. But I'm not privy to the timelines of the FDA. So
Okay.
we'll have to see how that works. But yeah, there's many different components to a BLA. So those are already underway, and they're in process right now. So believe you me, it's our goal as a company to move this along as quickly as possible, but we want to have the strongest package possible, with the goal of getting accelerated approval. So.
Yeah, I'd probably say it now rather than at a wrap up, but there is still an enormous unmet need in Duchenne. And anything that we can do as an organization to help support that and accelerate it, we will.
Thank you.
Yeah.
I know you're a wonderful partner for us.
Leanna, we got about 5 minutes for burning questions.
Um, No, I think that covers it. We actually had quite a few questions come in, and so we will pass those along to Regenx because there's way more here than I think we can answer in the time allotted. I think that speaks to all of the interest that everybody has. Of course, individuals are always wanting to know when will this be available to me. And I know since we have so much interaction with you guys at Regenx that you are also thinking about the older individuals, non-ambulatory, and looking to see when you can get there as well.
100%.
Okay.
Yeah.
Well, let me wrap it up and
Well,
we'll
I was going to say one more thing.
Sure.
The last question that just came in was that they were asking Naz if you were going to be at the Cure Duchenne conference, which I know we're not going to see you this week at Cure Duchenne Futures, but many of your colleagues will be there.
Oh,
So,
yeah. Oh, I'm sorry. I'll be missing you guys this
Yeah.
year.
Yeah.
Sorry.
I gotta say also that we don't have a ton of time at the conference to ask questions but of the presenters, and this has been a great opportunity to roll it out and ask questions and get feedback for the community. So I've appreciated that.
Sure, yeah, anytime. And my colleague Koji Takahashi is going to be there. He's amazing. He's super knowledgeable. He's wonderful, very approachable. So, and Vivian will be there from patient advocacy. So, and the rest of our medical affairs team, they're so lovely. I've been working with them, and I'm so impressed. They're just wonderful resources. We are all extensions of each other. So, you'll have a lot of really good resources there.
Okay, cool. Let me wrap this up. So I want to thank everybody for joining the Cure Duchenne webinar today. And obviously a big shout out to the whole team at Regenxbio for the work they've done in developing 202 and for sharing this a really important corporate update with the community. The results are extremely encouraging. And for me, it's particularly reassuring to see a statistically significant correlation between RGX-202 microdystrophin expression and functional improvement. It's what both scientists and families are looking for. We look forward to further updates from the company, especially as they engage with regulators. The unmet need in Duchenne is still there, still significant, and the company's focused on filing, as you've heard, a rolling BLA this year and potential for accelerated approval in 2027. We'll send out a post-webinar email to everybody to all attendees tomorrow. We'll let you know whenever it's posted online for viewing and sharing. And thank you, everybody, for joining. Have a rest of a wonderful day.
Thanks for having me and us from Regenx.
Yeah. See you guys.
All right, take care. Bye.